in the Graduate School of Education and the School of Environmental and Biological Sciences at Rutgers University. She received her PhD in the Learning Sciences at Northwestern University, and she currently has two main research strands: designing and studying of inquiry-based learning environments in the life sciences that engage students with modeling and argumentation, and studying learning progressions in science education, specifically in genetics. In addition, Dr. Duncan also coordinates and teaches in the certification program in biological sciences at Rutgers University, and has studied the development of pre-service teachers' knowledge and beliefs as they progress through that program. She is the recipient of several federal grants and has published in science education and teacher education journals, including the Journal of Research in Science Education, and Journal of Science Teacher Education. It is my absolute pleasure to introduce Dr. Ravit.
Thank you. Great to be here. And I'm going to just get started. I'm probably going to talk for about 45 minutes, and then we'll have time for questions, okay? And in terms of what I want to talk about, I want to talk about the development of the learning progression in genetics, the development of assessment items and instruction. Oops, I don't actually need that. And a study that actually explores sort of two genetics learning progressions, some initial results, and then what we're up to now, what we'd like to do in the future. So without further ado, learning progressions. So what are they? Learning progressions are descriptions of successively more sophisticated ways of reasoning about core ideas and practices in the domain that occur over long periods of time, okay? Grades and grade bands. And they're featured prominently in the framework for K-12 science education, sort of the backbone of the standards as well. There are several appendices that deal with learning progressions for core ideas and practices and even the cross-cutting concepts. And they're an attempt to bring greater coherence and alignment to standards, curriculum, and assessment. In science, learning progressions have four core characteristics. One is an attempt to cull the herd and really focus on a few powerful and generative ideas and practices in the domain. These progressions begin with a lower anchor, which takes into consideration students' prior knowledge, and end with an upper anchor, which is based on societal expectations for what students should know at that point, as well as analyses of the domain. And then the meat of the progression are essentially steps or levels that link the lower and upper anchor. And these are derived from research on student learning, extent that it exists, okay? And importantly, these are not developmentally inevitable. Rather, they require carefully designed instruction in order to foster student development along the progression. These are conjectures, okay? These are hypothetical conjectures about learning paths, and they need to be refined and iterated and studied. And that's part of what we're doing here. And this is an extensive process, okay? This is multiple years and rounds of revision. The genetics learning progression is grounded in a framework for genetic literacy by Stuart, Carty, and Passmore in 2005. And they have argued that genetic literacy entails understanding three interrelated models in the domain. The first is the inheritance model, and that describes inheritance patterns, right? Those are the correlations between genes and traits, right? We remember the big A, little a, and the phenotypes. The second model is the meiotic model, and this explains how you actually get the genes from one generation into the next generation. And the third model is the molecular model, and that model explains how a gene, once you have a big A or a little a, how does that bring about the traits within the individual? We've also added the component, or emphasized the component of environment and interactions between genes and environment to this framework. And then we identified 8 core ideas that sort of map onto this. And they map onto these models and the relationships between them. And in thinking about these ideas, this is what we wanted these core ideas or big ideas to be about. They need to be something, ideas that are important, in fact, for scientific literacy, for sort of civic engagement in genetics. They need to be powerful generative toolkit, right? They should allow kids to reason about genetic phenomena, not only the ones they're learning in the classroom here and now, but also genetic phenomena that they may encounter outside of the classroom and in the future. They should provide the basis for future learning. And another aspect that we had to deal with is this balance between fidelity and learnability. So the progression, even though it ends at sort of the end of high school or compulsory education around, say, 10th grade, the ideas in the progression are not the cutting edge science in molecular genetics, okay? Rather, there are, as I said, these powerful ideas that form the basis for future learning. And they're accessible. They're something that could be learned over time for learners. So we organize these 8 ideas into 2 sort of general questions. One is, how do genes influence how we and other organisms look and function? And this has to do predominantly with that molecular model. Okay, once you get the gene, how does it bring about the trait? And the second question is, why do we vary in how we and other organisms look and function? And this has to do predominantly with that inheritance model, the meiotic model, and part also with the environment. And this progression was developed by myself, Aaron Rogat, and Anat Yarden. It was published in 2009, at least the first version of it. So if you think of the progression, okay, we have our 8 constructs, our 8 big ideas. We have 4 levels of growing sophistication. So each cell essentially describes the expectation for that construct at that level. And the idea is that students develop all these 8 constructs in some form from the beginning of the progression and on, okay? So we're not necessarily arguing that one model needs to come before the other, but rather that they're all accessible in some form early on, and we get progressively more sophisticated. And what does that actually mean? One is deepening the understanding of each model, deepening understandings of the connections between these models, and developing these mechanistic explanations of genetic phenomena that start from sort of the macro in the earlier grades and move on to the micro and molecular in later grades. So just to give you an example of what one of these constructs looks like. This is construct B. It's one of our favorites. And it describes the idea that genes are essentially information for proteins or recipes for proteins. And at a very, and this is looking down towards up. At the very beginning, there's this sort of notion of a level 0 that kids don't see genes as information, right? They see sort of as genes as these passive particles or associated with traits. You get the gene, you get the trait, almost synonymous, okay? There's no mechanism here. They do know, though, that children and parents are related, and there is something that sort of parents give to kids, but it's not a mechanistic idea. They're not very sure about what and how. At a level 1, we have this very important shift from seeing genes as sort of these passive particles to seeing them as units of information. Now, at this stage, the units of information are for pretty much everything about you, right? It's kind of like a novel. It tells everything about me, how fast my heart's going to be, what color my hair is going to be, how tall I'm going to be. Any trait that I have is somehow sort of written in this blueprint. At a level 2, this becomes a little more refined and constrained, and now genes become information that's specifically for proteins. So no more information for just everything, but rather an understanding that all you need to specify in the system is the proteins, and then they just give rise to the traits through interactions and so on and so forth. And this is really rather counterintuitive. And lastly, this is essentially understanding the genetic code at the molecular level, understanding that 3 nucleotides code for 1 amino acid. And these amino acids make up chains that fold in particular ways, and voila, you have a functional protein. Now, if you're working with learning progressions, like it or not, you're going to be doing a lot of assessment. Okay? So in order to figure out where students are thinking along this progression, in order to develop a validity argument for the progression, you need assessments. And this is the sort of assessment triangle for knowing what students know. And the idea is that you need some sort of model of cognition, In our case, it's the learning progression. Then you need some observations because we can't read their minds, so we need them to do something so that we can get a sense of how they're thinking. And that's items, and there are various formats from items, from interviews, tasks, to multiple choice, to open-ended. And lastly, you have some sort of an interpretation model. This is usually a statistical model that allows you to link inferences from the observation back to the learning progression. All right, talk about that piece last. But the next thing I'm going to talk about is the assessment and development of the items. So if you think that you need about 3 items per level per construct, that's a lot of items, okay? So there's some issues around testing time, fatigue, and just teachers also not letting you take up that much time. There are also issues around contextual factors of items that can interfere, and I can talk more about that later. And you also want to make sure that the items are sort of accessible in terms of readability, graphic. You don't want to be assessing a whole bunch of latent variables and skills that are not part of what you're trying to assess. So we made a decision fairly early on that we're going to go with these things called ordered multiple choice items. These are essentially multiple choice items in which the response options are not just random distractors, but rather they map onto levels of understanding. Okay? They're great in the sense that they give you more information than traditional items, okay, multiple choice or even open-ended. They are challenging to write. They do kind of put a bit of an onus on the student. They have to select the most accurate answer, and sometimes your lower levels of reasoning are not entirely wrong, they're just sort of incomplete, and students may pick them over the most correct if they're not really reading carefully. So that is something that's a bit of a challenge as well. And of course, they require a really intensive process of validation in which we engaged. We decided, in order to make life reasonable, that we're only going to focus on 5 constructs. So everything you're going to see from this point on is only 5 constructs from the progression. 3 deal with molecular, 2 deal with sort of the meiotic and inheritance, the inheritance and meiotic model, what we call the classical genetics. Let me give you an example of an OMC item. This is an OMC item for construct B, the one I just showed you. DNA provides information for which of the following: structure and function of a protein, a trait that an individual inherits, assembling amino acids into protein molecules, or assembling protein molecules into amino acids. And if you level this item, or if you look at it, actually, let's look at it as an MC item first. This is the correct response. When we did a pilot with high school students, about 300 of them, after they learned genetics, using their own traditional instruction, only 23% of the kids got this right. These are 11th graders, okay? Not looking so great. But if we level the item now and say, well, but this is a level 3, this is a dot. Oh, nothing, just a regular distractor. This is a level 2, meaning they understand that it's information and it's for proteins, but they don't know exactly what's in the protein. Right? Level 3 is exactly what's in the protein. And a level 1, remember, this is information, but for the whole trait. If you look at it as partial credit, this is what you see. So, it doesn't change the numbers of the correct. There's still only, relatively few that are level 3. But now I have a better sense of where the rest of the students are falling. And we can see that most of them are not going, for the dot, there's level 2 and level 1. And so as a teacher, I have a sense of where my class is and what moves I might need to make next in order to move them more towards a level 3. Right? So in order to test our items, we conducted 3 rounds of piloting, one with kids that are 11th graders, essentially in the target district that we were going to work with for the study a year before the study started. Another with college students, because we wanted to make sure we were getting to the upper levels of the LP and the kids weren't sort of grounding out. And then we did it again in a different high school with high school students and AP students. We had teachers look at these items. We even consulted with a literacy coach to look at some of these items. Each of these rounds entailed a lot of statistics and figuring out which item is working and not. And this is what we ended up with. We ended up with a pool of 84 items, of which we used about 56 to generate 2 forms that were 45 items each with a 30-item linking block. That means it's identical items, same order on both tests. And overall, this is the coverage for the construct. So a minimum of 9 items, maximum of 13. So that was a little bit of the assessment. I'll get to the third part of the assessment later. But as I said, these progressions are not developmentally inevitable. So if you want kids to move, you need instruction. So that was our next piece. And for the instruction, we developed 2 modules. One that hit those molecular constructs, A, B, and C, and another in classical genetics that hit the E and F constructs. This was a model-based inquiry curriculum, meaning students engaged in modeling and argumentation in order to develop the knowledge, these core ideas. That was usually followed by benchmarks in which they sort of learned the respective terminology, practice, had some practice problem, and so on. And there were a whole slew of phenomena that we worked with, some sort of regular traits, hair texture, HIV resistance, and a set of genetic disorders. And just to give you a flavor of what this looked like from the student's end, these are 3 models. These are what we call rule set models that we gave students in the classical genetics module. So these are sort of hypothetical set of rules that kids their age, supposedly in a different class, came up with. One of them is correct, Sarah's model. The other ones are not, different in different ways. And the students had to look at these models. Then we gave them a set of evidence. And the epistemic game was to figure out, well, which model is best supported by the evidence and not contradicted by any of the evidence. So the evidence that we gave them, these are just thumbnails. It's not the whole evidence. But our pedigrees, we gave them some information about difference between egg cells and body cells. And this sort of anecdote of Mike, who is a college student, and he tells his sister or brother what he thinks is going on and how traits are inherited. So students use these pieces of evidence and they write an argument in support of the model that they chose. Similar kind of structure for the molecular model. Here, the driving question was, how can a mutated gene cause HIV resistance? So it's HIV resistant phenomena. And we gave them 2 competing models and a set of evidence. Again, these are just thumbnails. There's more to it than that. And what they had to do was relate the evidence to the models. We have a set of scaffolds that support them in doing that. And then they write an argument. Okay, so that is some of the main kind of activity structures in here. All right, so let's complicate stuff a little bit. So I told you that there was a genetic learning progression that we developed. Well, there's another one that was developed by Golan and her team at AAAS. And the main differences between these 2 progressions have to do with sort of the assumptions around the strength of cognitive constraints. So Golan argues with her progression that you really need to understand the molecular model first, because that's essentially the mechanism. And if you understand the mechanism, then the big A, little a actually makes sense. Otherwise, they're just abstract notations and kids don't really understand what they are. They can do the patterns and they can do the Punnett squares, but they're not really understanding what these represent biologically and what they're actually manipulating. As I said, in our progression, we were somewhat agnostic. We didn't say that either of these need to come first. Obviously, if you're teaching at some point, one of them does come first because you got to start somewhere. So to simplify this and make it a little more easy to study, we decided, well, what if we sort of did say that our progression assumed that you have to start with a Mendelian model, and Roseman's said the other way. Well, if the cognitive constraints are strong, meaning if you really need to put the knowledge in a particular way, and it is more effective to put it that way, and it's more stable to sort of build it that way, like a Lego tower, then if we build instruction that maps onto these progressions, one of them might be superior, right? The kids are going to learn better. So we had 2 research questions with this project. One had to do with the sequence, so which one is better? And the other had to do with the modeling. Is it the case that maybe one sequence is better for learning genetics but the other one is actually better for engaging in modeling and moving along a modeling progression. So I'm not going to answer the second question today. I'm only going to answer the first question, and probably only partially anyway. So this is the study design that we used. I told you that we had these two modules, so we could flip them. These modules were fairly equivalent in terms of time, kinds of modeling activities that students engaged in, number of benchmarks. They were sort of fairly, we tried as much as possible to make them structurally as equivalent as possible. We also had a couple of days in which students learned about modeling and argumentation with some activities that we have that are not specific to genetics. And then we had about a week's worth of what we called the bridge unit. And the bridge unit, the whole point was to help them connect between the molecular and the classical. And we had our content test, so we had a whole bunch of assessments. I'm not going to talk about all of them, but we had our OMC pre and our OMC post over here. We also have interviews, which we're currently analyzing, and a whole slew of other modeling assessment. But this is the general structure. And these classical, molecular, classical, those were within teacher, okay? So we had about 5 teachers and their students. Some of these were in honors, some of these were in lab classes. And for each teacher, we tried to balance both honors and labs and the classical molecular, which came first. We call them the MC, molecular classical condition, or the CM, classical molecular condition. So there are about almost some very similar numbers of CM and MC in this little school that we work with. So the analysis was conducted by my wonderful ex-postdoc now, Jeannie Choi, and Jeannie is an assessment and measurement expert, and she used a multidimensional IRT response modeling using software called Conquest. And you get a whole slew of outputs that have to do with item fit statistics and various other measures. I'm going to talk about some of them, but not all. So this is a right map. This is one of the generic kinds of representations that you get when you do IRT modeling or outlining progressions. And the nice thing about a right map is that it allows you to put items, these are item difficulty, and students on the same logic scale. So what I'm going to do now, though, in order for you to be able to see something in here, is I'm going to separate it. I'm going to talk about the item side first. So this is a right map. What you're seeing here are the 5 constructs, okay? Now remember, each item could be scored at different levels, right? Because they were ordered multiple choice. The reds are what's called Thrustonian thresholds for items at a level 1. So this is sort of essentially a 50% probability that a student will answer at level 1 or above. The greens are 50% probability student will answer at level 2 or above, and the blues are level 3 and above. And if we look at construct A, which is the prettiest one I have, and make it a little bigger, let's look at one item and see how it behaves. So let's look at item 5. This is where the threshold is for level 1. This is a threshold for level 2, and this is a threshold for level 3. So first, they're not exactly equidistant, which is fine, not necessarily expected, but they do go up. And that's true for all our items. So we didn't have any reversals of, a level 2 that's supposed to be a level 2 looking like a level 3 or a level 1. So this goes up. That's true for the other constructs. One of the things that you'll notice with the construct is that there's some overlap, okay? If this was an ideal world, you get a nice red band and a nice green band and a nice blue band. That's not exactly what we're getting. Most folks don't exactly get that either. Now let's look at the student side. So this is the overall gains. I know it doesn't mean much. I'll show you a little more in a second. We essentially got gains for all the constructs, which is great. At least the do no harm they learn. What do these gains mean? If we look at construct A and we look at sort of the A here, so the kids are coming in at, they're just about proficient at a level 1, okay? Most of the kids are sort of at or above here. They leave with sort of mastered level 2 and just sort of starting level 3. Okay, so it's about a level or so jump in terms of understanding. I'm sort of trying to think what this means. Here's another way to look at it. These are box plots. And what you see here is that the medians more or less fall on the same line. Construct B was harder, right? So kids came in knowing less. They learned a lot, so the gains are big. This was one of the harder constructs. This is the same construct I showed you, right? With the idea of understanding the genes or information, what their information. It's counterintuitive. It's a hard one. They sort of had the smallest gains for construct F, but overall it's more or less the same. So they learned, and that's great. One of the questions that came up was, well, did they learn more than the traditional instruction? We didn't have a control, right? So this study did not have a control group built in. But remember, we did the pilots before we started the study. Study was actually 2 years. So we did piloting, got all the assessments and instruction and all done. And then we came into the school. These, again, were 11th graders. For 2 consecutive years, we modified the curriculum a little bit between year 1 and year 2, but for the most part, it was the same curriculum. Teachers got better at using it. So if we look here, this is the gains, overall gains for year 2. This is what the pilot was. Same kids, well, not same kids, same school kind of kids, but a couple of years before. So that was great. It's also good. Even in year 1, we did better than traditional, but there is a jump also between year 1 and year 2. But what really made us happy, or at least smile, is that remember we did the pilot with college students, right? These are about 100 level kids. This is after they learn genetics in the regular 100 level. And we do as well or better than that group as well. So that's nice. We also looked at correlations between constructs, and what we find is that the molecular constructs are about moderately correlated, which is what you would expect. They kind of hang together. They're less correlated with the classical constructs. In a second, though, you'll see something else. The classical constructs are really not that correlated, which we're not entirely sure why. And funnily enough, construct B and construct F are correlated. Some level, this is a bit of an artifact. Why? Because construct F is that relationship between genes, like your genotype and your phenotype, right? Well, at a level 3 of that construct, what we want students to be able to do is tell us what those genes code for, right? What is the story at the molecular level for why big A, little a is healthy if you have a recessive disorder, but you're sick if you have a dominant disorder. How is that possible? What's a molecular explanation for that? So that's what the upper levels of level of F are, and that's why you technically need construct B to really do well. So that's probably why there is a correlation. But the thing that you've been waiting for is like, okay, well, what about the MCCM differences? Did they learn better in any of these sequences? So no. There's no statistically significant differences between the MC and CM conditions, which was not terribly surprising. We weren't hanging our hats on this, but there is something interesting. So if you look across the board, for all of these constructs, the red, which is the molecular first, comes up a touch above. Now, you would expect, if there's recency effects, that the kids at the molecular first would do better on the classical items because they just finished them, whereas the molecular, they like 6-7 weeks ago. And that's not the case. So, I mean, that is the case. They do better on E and F, but they also do better on those constructs that are the molecular constructs that they learned quite a while ago. So the fact that this trend is consistent across is what's sort of curious here, and suggests that maybe there is something to be said about starting with molecular. We also gave the post-test after we did the bridge. Now I'm wondering if we'd given the post-test before we did the bridge, if this would actually have been more striking of a difference. Anyway, so conclusions. So like I said, there's no statistically significant differences, but there's this trend that favors the molecular first. which made Joellen very happy when I told her that last week. Students gained overall, they gained the most for construct B, mostly because they came at a lower level. They gained the least in construct F. We actually know now from research that Moraima has done that construct E is harder than F. So we're actually now looking at the relationship between constructs. That's some of the stuff that we're working on right now. But we do that mostly using interview data. We know that the molecular constructs are correlated. They're more correlated to themselves, less correlated to the classical, and the classical ones are not that strongly correlated. There's still some issues with these OMC items, so we're still getting a little too much overlap between levels 2 and 3 for most of the constructs. So what are we going to do? Oh, I should have fixed this slide. It's old now. But so if you did go to NARCS, you would have seen Veronica talking. We're looking at the relationship between content and modeling, and also in terms of what kinds of argumentative strategies students use in constructing arguments. So that was Veronica's talk. We're looking at relationship between constructs. So when we developed the progression in 2007-8, there wasn't enough literature to make any sound conjectures about which constructs might need to be learned first. Or is it the case that, for example, you need to be at a level 2 on B before you can be at a level 2 on C or something like that? So that's something that we're exploring now. And we're also engaged in a cross-sectional study, which we're going to get ready to launch hopefully this week, that will look at from 7th graders, middle school, high school, college, and take it nice and sort of into college, like graduate students, to get a handle on what's happening at that upper level of the progression. And that's pretty much it. We're funded by NSF. As I said, I'm thanking Ginny for the wonderful analyses that she's done. And if there are any questions, I know. Yeah.
So I guess I'm unclear, just to talking about the future directions, but also it just seems like the test you did comparing the alternative learning progressions is a test about relationships between constructs. It's not about individual constructs.
It is. So those are all individual. This is all individual. This is A, right? This is overall. So overall, the ones that did molecular first come out on top by a little bit. Not statistically significant, but a little bit. And for each and every construct.
Right. So your data is about each and every individual construct, but the idea, like the overall test, is a claim about whether some constructs come first or
no, not so much. So the items are designed for constructs, right? So each item assesses at least 2 or 4 levels, or between 2 and 4 levels of a construct. So we don't have any items that look at relationship between constructs, not for the OMC items, okay? Our interview data, which is what we're using to develop the relationship between constructs, that's richer. And we've been using that to try and get a handle on what do they seem to know first, or what do they need to know first in order to learn other things. So this test does not look at those relationships. This test either gives you overall gains or by construct, but it doesn't look at relationships.
I feel like I'm not being clear, but so yeah, the assessment doesn't do it, but like your comparison of your LP versus the Wilson one, is that fundamentally a comparison about whether constructs are ordered in some way, like different constructs, not within a construct, but across constructs.
Yeah.
And how does, I guess my question is, how does an LP specify relationships between constructs, not just levels within a construct?
So you're right. So if we're arguing the molecular should come first, then those constructs that are associated with that model should be taught first, which is essentially what that instructional sequence did and what you're seeing here. In terms of, but the OMC items can only speak a little bit to that, right? So this is about as much as you can sort of get from them. We don't have enough items to see if students who score at a particular level on one construct, how did they score on another level of a different construct? I just don't think we have enough items for that, although I do need to think about that. Maybe there's a way to do that. And the way we're trying to get at relationships between constructs, including molecular to Mendelian, is through the interviews. And to try and get a handle on what they seem to be understanding and how that helps them explain other things. Now, we only did a pre or post interview, so we don't actually have an interview in the middle, which would have been really great. There's just no time. So we're trying to get an answer to that. There are lots of different ways in which constructs could be related. And it's something that sort of work that's ongoing. We've identified some relationships between construct B and C with the paper that works up that Nicole Shea did a couple of years ago. Now we're looking at ENF. We need to look at ENF and relationship to the molecular. So all of that is something we're still working on. That's the long answer. Yes.
This is a great presentation. Thank you so much. And it's great to see what you guys are working on in science. So I like your presentation because it shows how complex learning progressions really are. And you did it for science, but I know for math, it's the same type of thing. They are so complex to try to get your mind around. So my question is about the beginning of your presentation when you laid out the learning progression ideas. Is any of that literature that you reviewed, that you mentioned in your presentation, or other stuff that you reviewed about learning progressions generalizable to other content areas, or was it specific to science? And then a sort of related question is the type of question, when you described how you designed your questions, is that a generalizable idea, or again, is that particular to science?
So I'll start from the end and we'll go. So I think OMC items you can do across the board. I don't know of anyone who's done OMCs for math, but I can't imagine you couldn't.
Okay.
I actually think Jimmy's work, isn't Jimmy's work kind of around that? He doesn't order them.
No idea.
Okay, so Jimmy looks, my understanding what Jimmy does, Jimmy's not here, so I guess I just have to deal with what I understand Jimmy to be doing. Jimmy looks at, let's say you have 5 skills that you need to solve a particular kind of problem in math. He's trying to figure out which skills you need and how that sort of progresses. So he's not ordering them right off the bat. He's sort of seeing, it's a little more like the facets, like Jim Minstrel's facets is the way that I think about it. So, so there is, and that's in math. So I think you could do this in math. I don't see any reason why you wouldn't be able to use OMC items in math. In terms of the literature, so obviously the studies that we looked at, they were about genetics, don't generalize, they're about genetics. Literature on learning progressions at large, there are quite a few articles that try and sort of develop a taxonomy of learning progressions and the different kinds of progressions. And actually, there's some interesting differences between the math trajectories. I don't call those progressions, right? Mm-hmm. So the math trajectories, which have come before, they have a longer history, and learning progressions in science. And there's sort of 2 key differences that I see. One is that the math trajectories are grounded in teaching experiments more so than the science one. It's not the case that none of the science ones are grounded in teaching experiments. There are some, like Rich Lair and Leona Schobel's work certainly is. But Andy Anderson, for example, who has a carbon cycling progression, he developed that one based on cross-sectional. And is now moving to teaching experiments to develop validity arguments, right? So, but the math ones, I think, were more grounded in starting with the classroom. Those trajectories also include information about instruction. So there's a large sort of debate in the science progressions. Do you need to specify instruction on how to move kids between levels? Or is a progression just a theoretical sort of model, cognitive kind of model? And some people say, yes, you need the instructions, kind of useless without it. And others are like, but you need it, but it's a separate piece and it's not part of what we call a progression. Whereas the math trajectories, I think, have that a lot more built in to the trajectory, like the CGI work. So there are some articles out there that are generalized and can sort of help you think about a progression.
Thinking about the sort of what is happening at the instructional level, the teachers that you worked with, did you engage them in some kind of professional development to kind of prepare them for using the modules you developed, and how
Yes.
intensive was that?
Well, so it's very intensive on our part. So what happened, so this is what happens in a project, right? So I proposed to NSF that we'll work with 4 teachers. We'll get them early, like 2 years before the study starts. We'll do an intensive summer. Then we'll work with them throughout the year before they even get to the kids and the materials. We'll work with them in developing the materials. And then we'll have all these 4 teachers work with us through one study and through a second year of iteration. We got to the school, they're like, well, we have 6 teachers. All right, fine, we'll take 6 teachers. We don't want to leave anyone out. So we did 6 teachers. We gave them intensive summer PD, worked with them. Beginning of the year, one of them went to teach chemistry. And then they told us, well, there's actually more teachers, and if you want to work with us, since we're all having the same test, you need all the teachers. So you got more teachers, some of which did not really want to be on the project. So we worked with them, but they didn't have the summer, so we kind of had to do all that. And then we came to sort of year 1, we had a group of teachers, and then for year 2, we had a bunch of teachers leave again. One went to teach AP, it was no longer part of the study. 2 new ones came, one left for a different district, and a new. So we ended up having 3 teachers that were there in year 1 and year 2, which ended up being, 2 of them ended up being our focus teachers that we videotaped and worked with. So yes, we did give them a lot of PD. Part of it, though, wasn't quite as effective because we gave it to the wrong people at the wrong time. But we were there, we worked with them, we were in the classrooms, we did PD sessions throughout the year to talk about some of these. I have to admit that there was huge learning curve for the teachers in that year. I mean, the reason you see the differences between year 1 and year 2 in the gains, that's the kids came in a little bit higher too. So the kids in year 2, the pre's were a little higher. The group was just a little more motivated and a little less contrary than the group we had the year before. But the teachers also bought into it, understood it better, made it their own that summer. What we actually did is that we're like, okay, we know there's some things about this you don't like, it's too long, fine. You take it, you fix it, you run it by us. Instead of us making it, running it by them. We sort of flip, but we couldn't have done it in year 1 because they couldn't have developed these kinds of modeling activities. So we did that. So yes, there was a lot of PD, and I'm sure that had we done it a 3rd year, we would have gotten even more gains because they would have gained facility with. These are really ambitious instructional practices.
Right.
And the group that Clark and I work with, the Praxis Project, where this intensive PD, it's you can move teachers, but it really does take a lot of work, and it has to be grounded in classroom work around curricula. This is not the kind of stuff you can do a 1-day workshop on, which we know. Yes.
So if I understand learning progressions correctly, it's not just the idea if you do a bunch of stuff in a year, you're going to get gains. It's the notion that there are contingencies among the order, that it really is good to do it in this order. And it seems to me that your results call into question the results of many teaching experiments' claims about showing that a particular order is good, because you've basically shown that 2 orders are good. So other people who are not doing what, because I think your study is really unique in testing 2 different orders against each other. So it makes me wonder whether what I should take from teaching experiments that show gains, because yours suggests, well, maybe somebody else could have come up with a dramatically different sequencing and gotten the same gains.
So, right. And that is an argument that's ongoing, right? So there are some people that are sort of saying, well, I mean, if that changes in sequence will make a large difference. Part of it has to do with this notion of how strong the constraints are, right? So Marianne Weiser, who's got a progression around sort of for young kids, early understandings of sort of matter and properties of matter. And they argue that these constraints are really, really strong, meaning if you get the order right, it works. And if you get the order wrong, it just doesn't work. They don't really learn. They don't really understand. It sort of doesn't stick, I guess, to use a colloquial word. And they've actually shown with teaching experiments where they had a control that kind of did it the other way, that and they did it across 2 years, that their sequence was really quite superior. And they had specific reasons for it. But the other was not sort of an alternative progression. It was just sort of the regular status quo, which doesn't really take into account research on student learning necessarily. So yes, I mean, and like Amelia Gottwals and I wrote about that in like Science Hour, a little focus piece that pretty much said, it's like, well, if you really want to know, then you need to kind of do these comparisons. Otherwise, you can't really tell. The thing is that genetics has more than one progression out there. There are a lot of other domains for which there isn't another progression. So there are no alternative or sort of competing arguments for sequencing. So I don't know, but I think that this does question this notion. I mean, the other issue that you didn't quite mention, but is a problem. So I said in the beginning, if you were paying attention, that these are multiple grade and grade bands. That's not what we were doing. We were doing it in one grade. How's that possible? So that's something that a lot of people do. This larger progression, but then you test it in a particular grade, mostly because doing longitudinal studies is incredibly hard, difficult. I actually tried that. I can tell you that story is interesting for other reasons. What we did see though is, so the progressions assume, our progression assumes that if you learn certain things in 5th and 6th grade, you come into 7th grade knowing more, you move on to 9th grade knowing more. Well, it turns out that our high school students were pretty much coming in with 5th and 6th levels of understanding. So it didn't take years to move them with what we would have considered middle schoolers had they gone through the progression. So we did move students across multiple levels within 1 year, sort of a speeded up version of the progression. So, I mean, there are also arguments about, is that legitimate to do this if we're supposed to be really doing it over extensive periods of time, and if you take into consideration that there might be connections between progressions across other subjects, like what they know in chemistry, what they know in math, and other that might also interfere here. So to really study these, you would need a lot of money, large groups, longitudinal study, none of which are currently entirely funded. But you got to start somewhere, right? I mean, you kind of got to have a starting place. Yeah.
Yeah. For your research design, you mentioned the students are assessed by writing argumentation.
Mm-hmm.
Yeah. So does argumentation also show these kind of learning gains?
So we're sort of not quite there yet, right? Veronica is working on that. You can ask her. So we are looking at argumentation. So we have multiple kinds of modeling and argumentation assessments. We have one which we call sort of content-less, even though it's not, it's in biology, but it's not about genetics. It's our general modeling assessments. Then we have genetics modeling assessment. We have one in molecular and one in Mendelian. So Veronica has analyzed the molecular ones, and we're starting to analyze the Mendelian ones. And for those, we don't have a pre, because there's only so many hours of the day that we could have asked these teachers to let us test the kids. So we don't have a pre for those. But we could look at the general modeling, which we haven't yet analyzed, which now Maya's going to answer right there. She's going to analyze those. And one could look at gains across that. We can also compare that to the middle schoolers that Clark and I work with, because it's essentially the same assessments. We use the same assessments across multiple projects, which can allow us to do all sorts of interesting comparisons. But that is all up and coming work. It's a lot of work. So if anybody is looking for work. Yes.
So thank you for sharing this. It's wonderful to hear. And I'm wondering, in terms of the reach of the work once you leave the district, say, or the school, are there recommendations that folks have taken up from you that you've made, sort of concluding from this research, and then that they've taken up from you in terms of how they adopt textbooks, how they sequence instruction?
So the district that we happened to work with happened to actually be doing molecular first even before we walked in. So that was actually the way they were organizing, right? I think, yeah, they did molecular first anyway. So that's great, right? That just sort of suggested that this may well be the more reasonable way to do it. Again, our evidence there is not the most conclusive, but so when we started the work, the NGSS was in the womb. It was there, it was developed. The people that knew what was going on knew this thing was coming. But the teachers and districts were a little more like, well, let's see. When we left, the NGSS was essentially adopted. And as of next year in this fall, it's basically go for middle to high schools. Students and teachers. So initially, some of the pushback we got is like, well, this is nice, but their tests, by the way, did not have any modeling in them at all. And they would actually argue that it's not fair to test modeling because what if students weren't good at it? We're like, but. So they didn't really see the value in the practices and focusing on the practices, at least not all of the teachers. By the time we left, I think that became more apparent. And I think now it is apparent, and I don't know exactly what materials they're using. I haven't gone back and asked, what using our lessons? I think they're using some of them. I know they're trying to build things that emulate some of what we've done. So we'll see, but these are really difficult to build. I mean, these are materials that actually Clark and I developed in some form even before the high school study. So I really co-opted a lot of the materials that we had developed for the Praxis project. And that took years. I mean, it's a massive development project. So I don't really see how teachers can necessarily do all this without heavy support and time. I mean, I need to go back and check, see what they're up to.
But in a sense, Ravit, aren't you trying to help them with a paradigm shift in thinking about science teaching as relaying a bunch of facts to engaging in practices of reasoning and modeling? And, and that's probably, the biggest culture shock for science.
Right, and it was not just for the teachers, it was students with the parents. I had to go talk to parents and explain to them that, what you want from your kid is to be able to reason, right? Some of them actually don't necessarily want that for their kids, which is surprising. But they're like, well, we just want her to get an A. And she got an A before you came on. And now she's not getting an A anymore. So that, I mean, that was a problem. I mean, the kids were used to worksheets. They were essentially PowerPoints that had lines. So the teacher would show like a PowerPoint and this, and imagine that instead of this, I'd have a blank line on your version. Mm-hmm. That's what the kids were used to. That's pretty much that, and some labs. Pretty much what they did. And we come in there's no more lines. And it's worksheets, and it's writing. A lot of writing. And reading. A lot of reading, like whole paragraphs. And that was a shock. And the kids rebelled. And the teachers are like, whoa, we're having all these I mean, we had a crisis in year one around like November, where it's like, or not November, later on the year, we had a crisis at some point in year one, where the kids were not motivated and the teachers were like, we don't know what to do with the complaining, and they're not completely bought in, so they're not really actively doing things around the complaining that would have necessarily helped. And year two was way better. But yeah, I mean.
Yeah, Ravit, good stuff. Thanks for the presentation. This is actually timely for my pro-sem class. That they had just read some articles about design research and some design studies that we're going to discuss tonight. So building on your last answer, what some design researchers say is that design research involves looking carefully at the contingencies and making instruction work. And so what you just described is dealing with some pretty big contingencies, dealing with the student population, the teachers, and the surprising, or maybe it shouldn't, the parents. To what extent do you theorize about that, and to what extent is that going to appear in your reports?
So, we theorized, so we knew that the scientific practices are going to be new to students and teachers. And we did have aspects of the design, like those initial activities that were supposed to help kids come on board. And there are scaffolds in there that take into account the fact that the kids don't necessarily know that. In terms of theorizing around the teachers, I mean, if we write papers around PD, which I'm not necessarily sure that we will, because we didn't actually collect data around the PD, that would be some contingencies that we need to go in there. The parent story, I don't think, is necessarily going to come out. But in the reports, mostly the contingencies that we talk about have to do with students and how what we know about where students are coming from impacts the design and how we dealt with that. So we did have to do some things around motivation, which we could easily talk about in the report, what we had to do in order to lively and spruce things up. We ended up a ton of videos, for example, which might seem like minor little things, but they made a huge difference in terms of motivation. If you don't have the motivation there, then engaging kids in all this work, I mean, it's just not going anywhere. We also modified evidence. I mean, we sort of learned a lot about how to present some of this to students in ways that were a little more appealing and less off-putting for them, a little maybe more similar to what they were used to. We also came in year one. We started, when did we start? I think we started like around the snowstorms. So the kids had the whole worksheets in the beginning, and then we came and changed the game. In year two, we're like, no, we are coming in early. So we did. So they didn't get enough time to enculturate into that. There was one unit before, but then we came in. So I think that was also another reason there was less pushback. So we didn't change the game on them in the middle of the year after they'd gotten comfortable with the prior game.
One more question?
Thank you for sharing your research. I was just wondering, like, curious, like, do you have any students with special needs or disabilities? Like, is there collaboration?
So at the high school level, we had one class that had in-class support, right? How many? We had two that had in-class support. The materials were shared with the teachers, and the teachers shared it with in-class support. I mean, one of the things that we did in year two, and also in year one, based on feedback from the teacher, is to structure the materials in ways that would be more accessible to students, sort of trying to reduce the cognitive load of it. So it wasn't specific to any particular disabilities, but in terms of trying to reduce the cognitive load, to have different representations and different ways that students could access the same information. We work with the literacy coach, not only on the items, but also on some of the instruction. So we learned some stuff from him. Like the biggest thing was like white space, like the good's good, white space is good. So, which again is trivial, but you don't know that if you don't, you know, it's not something you just necessarily know. So I don't know how the special ed kids or the ELLs fared on this necessarily. Some of them did not consent. So, but we didn't know who they were. We just sort of know that the teacher sort of reported that some of them were not consent.
Again, thank you very much. And am I right in that you're teaching design-based research next semester?
I am doing DBR, so all y'all want to learn about design-based research, it's open, not just, it's really a required EDD course, but it is open. Anyone else? Do lots of fun.
Learning science.
It is a learning sciences requirement, that's right.
And we have 2 more brown bags, Adam Gamaran next week, and Cheryl Van Ness will close off the season for us. Thank you all, and see you next week.
Both wonderful sessions.